Good afternoon. Thank you very much for giving me this opportunity to present this webinar on gastroenterology and hepatology. I am Juan Sastre. I am professor of physiology at the Department of Physiology, Faculty of Pharmacy in the University of Valencia, Spain. And I'm going to talk on the regulation of intracellular cystine reduction and protein cysteinylation by TRP14, thyrodoxine-related protein of 14 kilodaltons, and its modulation of the inflammatory response in acute pancreatitis. We have focused for the last years on the regulation of cystine metabolism. Cystine has a great importance in the general metabolism, not only for protein synthesis and glutathione metabolism, but also for the regulation of protein functions through mixed disulfides, particularly through the formation of a protein cysteinylation. And also the redox pair cysteine with cystine, its oxidized form, has a very important role in the redox status in cells, but its regulation has not been studied, nor the regulation of protein cysteinylation. If we consider that cells uptake cysteine through specific carriers, but mainly through the XCT minus carrier with interchange with glutamate, then it is critical to know how the uptake of cysteine yields cysteine, and the regulation, the reduction of cysteine to cysteine was unknown. So it was very important to elucidate which was the enzyme, which were the enzymes responsible for this reduction of cysteine. In addition, if we consider the protein oxidation, the regulation of protein oxidation, there are many studies on the regulation of S-nitrosylation and S-glutathionylation, but there were no studies on the regulation of protein cysteinylation. That's why we wanted to focus to elucidate these two critical steps in cysteine metabolism: the reduction of cysteine and the regulation of protein cysteinylation. And for that, we used an experimental model in which we have been working for the last decades, which is acute pancreatitis as an experimental model of acute inflammation. One of the hallmarks of acute pancreatitis and general inflammation is glutathione depletion, the decrease in GSH. Without, in this case, I was surprised that this occurs without glutathione oxidation, without an increase in oxidized glutathione. And remarkably, in acute pancreatitis in the pancreas during acute inflammation, there is a huge increase in protein cysteinylation, which is almost undetectable under basal conditions. This finding prompted us to define disulfide stress as a novel subtype of oxidative stress. This disulfide stress is due to the hydrolysis of glutathione due to proteases that become active in acute pancreatitis. Then glutathione is hydrolyzed and its components are released, cysteine and gamma-glutamyl cysteine, and they form mixed disulfides, protein cysteinylation and protein gamma-glutamyl cysteinylation. We were the first to describe protein gamma-glutamyl cysteinylation. So this formation of mixed disulfides in absence of glutathione oxidation was the major characteristic of disulfide stress. Looking for potential candidates to regulate protein cysteinylation, we focused on the thyrodoxine system. In the thyrodoxine system, we have the thyrodoxine protein together with the enzyme thyrodoxine reductase and NADPH. And in the cytosol, the isoenzyme, which is responsible for the action of within the thyrodoxine system, is thyrodoxine 1. Thyrodoxine 1 is able to reduce not only peroxidoxins, but many redox-sensitive transcription factors. And it has a very important role in redox signaling. As an alternative, TRP14, thyrodoxine-related protein of 14 kilodaltons, was an alternative candidate because it also exhibits a disulfide reductase activity, but there is a difference between TRP14 and thyrodoxine 1, which is the presence of an aspartate residue in the active center. And then this enzyme has a different specificity, different selectivity for the substrates. So the typical thyroxine 1 substrates cannot be reduced by TRP14. So TRP14 cannot act on insulin, ribonucleotide reductase, or peroxidoxins, among other substrates. And TRP14 has a very efficient cystine reductase activity, and this was reported in vitro using purified proteins, but it was never tested in live cells. So we wanted to explore the role of TRP14 as cystine reductase in live cells and its role in the regulation of protein cysteinylation. And for that, we established a collaboration with Professor Elias Arner from the Karolinska Institute in Sweden. And this was the major part of the thesis of Pablo Martí, who performed most of the work. And when we incubated HEX cells with cystine labeled with a body pie, we detected a very intense cystine reductase activity in the wild type cells, but this activity was absent in TRP14 cow cells. And we follow the kinetics of this reduction of cystine, and we confirm that TRP14 cow cells, they didn't exhibit this cystine reductase activity. So we can ascribe the major cystine reductase activity of live cells to TRP14. And this requires its active center, mainly the cystine of the active center. So when in TRP14 cow cells, HEX cells, we overexpress the wild type form of TRP14, we rescue the cystine reduction. And if we overexpress a mutant with a different amino acid replacing cysteine, a cysteine mutant, then we lost some of this cystine reductase activity. So the cystine reductase activity is required for the cystine reduction in the cytosol in cells, in mammalian cells. And this has a profound impact on the cell response towards any stress. And in particular, among other targets, we found that NRF2 can be upregulated in vitro in TRP14 cells if they are incubated with a high concentration of cystine. So cystine promotes the activation of NRF2 in absence of the cystine reductase activity typically of TRP14. Also, there is an impact of TRP14 on cysteine metabolism. And to assess this impact, we established a collaboration with Professor Peter Nagy from Budapest, and with his expertise, we analyzed the flux through the transsulfuration pathway, the synthesis of cysteine from methionine by incubating HEX cells, TRP14 cow HEX cells with methionine labeled with 34 sulfur. And we measured incorporation of this 34 sulfur into cystathionine, cysteine, which is the end metabolite of the transsulfuration pathway. And when the HEX cells were incubated with low concentration of cysteine, then there was upregulation of the transsulfuration pathway. So increased incorporation of sulfur, the sulfur isotope into cystathionine. And also it was promoted even the synthesis of GSH. So the deficiency in TRP14 together with a low concentration of cystine in the medium would induce the transsulfuration pathway to promote the synthesis of cysteine from methionine, because this is another alternative source of cysteine for the cell metabolism. And regarding the regulation of protein cysteinylation, we did that with in hex cells incubated with biotinylated cysteine, and this was in collaboration with Dr. Antonio Martinez Ruiz from Madrid. When we incubated the cells with biotinylated cysteine, we found an increased incorporation of cysteine and increased protein cysteinylation in the TRP14 cow cells, meaning that the deficiency in TRP14 was promoting cysteine, protein cysteinylation. We discovered that the TRP14 system, together with TRP14 together with thyrodoxine reductase 1 and an ATPH, were able to reduce these mixed disulfides, this protein cysteinylation. And we studied which proteins become more oxidized due to protein cysteinylation upon deficiency of TRP14 to look for targets of protein cysteinylation upon deficiency of TRP14. And among these targets, we found the elongation factor 2 and, importantly, peroxyredoxin 2 and other peroxyredoxins. Peroxyredoxins are required for the removal of hydrogen peroxide. And we, using purified proteins and the thyrodoxin system, thyrodoxin 1 together with thyrodoxine reductase 1 and an ATPH, we demonstrated that this system is able to decysteinylate these mixed disulfides, protein cysteinylation. And the TRP14 system, that is composed by the enzyme together with thyrodoxin 1 and an ATPH, were also able to decysteinylate cysteinated peroxyredoxin 2 using cysteinated peroxyredoxin 2 as substrate. So we demonstrated that both systems, the thyrodoxin 1 system and the TRP14 systems, are able to decysteinylate cysteinylated proteins. The cysteinylation of peroxin 2 causes its inactivation. Peroxin 2 is able to detoxify hydrogen peroxide. And using the FOX assay, we showed that when peroxyredoxin 2 was cysteinylated, it became inactive, and we had to add TRP14 to recover the activity of the detoxification capacity of peroxyredoxin 2. So it regulates, protein cysteinylation may regulate the function of proteins, and in this case of peroxyredoxin 2. When we compare deficiency of thyrodoxin 1 with TRP14, although using pure systems, thyrodoxin 1 was more active than TRP14, when using complex systems by adding additional substrates, typical substrates of thyrodoxin 1, such as hydrogen peroxide or insulin, oxidized insulin, thyrodoxin 1 has a priority to its typical substrates and then exhibits less efficiency for the reduction of protein cysteinylation, whereas TRP14 maintains this efficiency for the reduction of protein cysteinylation in presence of insulin or in presence of hydrogen peroxide. Thyroidexin 1 loses this capacity to reduce protein cysteinylation in a pro-oxidant environment with high hydrogen peroxide, which is another substrate for peroxythyroxine 2. We also demonstrated that the role of TRP14 is maintained through evolution because it is maintained in other species. And for that, we collaborated with Antonio Miranda from Seville in Spain, who has a great expertise in C. elegans. And he has all the genetic tools to assess the role of TRP14 in cysteine metabolism. Well, he confirmed that the TRP14 ortholog in C. elegans, which is TXDC17, exhibits cysteine reductase activity. And when we established the genetic model of C. elegans deficient in the TRP14 ortholog, as well as in cystathionase 2, which is the key enzyme in C. elegans for the transsulfuration pathway to synthesize cysteine for the hydrolysis of cystathionine. So when in worms, when they are deficient in cystathionase 2, they have blockade of the transsulfuration pathway. Upon these conditions, if we add a proteostatic stress, which is due to a mutation, deficiency, specific deficiency in the worms, RAM-LS 133, that causes proteostatic stress, the accumulation of oxidized proteins. This causes the death of the worms. So the worms were not viable. When we blockade the two sources of cysteine, which is the TRP14 ortholog and the transsulfuration pathway through cystathionase 2, then the worms, they don't have any source of cysteine. And in presence of an additional stress, they become not viable. So this demonstrates the critical importance of providing sufficient cysteine to cope with additional stress. So it's critical to get cysteine either through the cysteine reductase activity of TRP14 or through the transsulfuration pathway. Then we move to the in vivo model in acute pancreatitis. And for that, we needed the TRP14 cow mice that were kindly provided by Professor Ed Smith from Montana University in US. And when we induced acute pancreatitis in the cow mice, we saw unexpectedly that there was no increase in protein cysteinylation. The protein cysteinylation, the disulfide stress that we found and we confirmed in wild-type mice upon pancreatitis, was not present in the cow mice. Why? Why there is protection against the increase in protein cysteinylation in this mixed disulfide? The mechanism was due to the upregulation of thyroidexin 1, the alternative mechanism to reduce protein cysteinylation in the cow mice with pancreatitis. There was upregulation of the mRNA of thyroidexin 1 as well as of the protein, induction of the protein. And paradoxically, the TRP14 cow mice, they were protected against pancreatitis, at least in part due to upregulation of glutathione synthesis. Glutathione is synthesized from cysteine, requires cysteine as a source, and it requires the transsulfuration pathway. And the TRP14 cow mice, they exhibited no glutathione depletion. The glutathione depletion that is present as a hallmark in acute pancreatitis was not present in the cow mice. Why? The reason for that was that the transsulfuration pathway was upregulated in the cow mice with pancreatitis. So the cystine depletion that occurs in pancreatitis in wild-type mice was not present in the cow mice, although these mice exhibited a remarkable increase in cystine, in the oxidized form of cystine, because they lacked the cystine reductase activity. To confirm the upregulation of the transsulfuration pathway, we measured the metabolites of the transsulfuration pathway by mass spectrometry, and we found that the remarkable depletion of homocysteine and cystathionine that occurs in wild-type mice with pancreatitis was not present at all in the cow mice. And one of the key regulatory enzymes in the transsulfuration pathway, which is cystathionine beta synthase, was upregulated in pancreatitis in the cow mice, confirming the upregulation of the transsulfuration pathway. Together with the upregulation of the transsulfuration pathway and the increase in cysteine levels, we also found an increase in the levels of gamma-glutamyl cysteine, the precursor of glutathione, explaining why glutathione synthesis was promoted and favored in the cow mice with pancreatitis. And one of the major regulators, key regulators of glutathione synthesis is NRF2, through the regulation of glutamate cysteine ligase, the enzyme that forms gamma-glutamyl cysteine. So there was a real basis to explain why there is an increase in gamma-glutamyl cysteine levels, not only for the increased synthesis of cysteine through the transsulfuration pathway, through the enhanced transsulfuration pathway, but also through the upregulation of glutamyl cysteine ligase. And for that, we needed to confirm the activation of NRF2, and we confirmed a remarkable upregulation, induction of NRF2, increase in NRF2 levels in the pancreas in cow mice with pancreatitis, and an increase in the nuclear translocation of NRF2, confirming its activation and increase of glutamate cysteine ligase. And the upregulation of the transsulfuration pathway, together with the activation of NRF2 and the increase in glutathione synthesis and the maintenance of glutathione synthesis, was very beneficial in the pancreas in cow mice with pancreatitis. So the cow mice with pancreatitis, paradoxically, it was protected against tissue damage in acute pancreatitis. You have here the remarkable increase in tissue edema and in inflammatory infiltrate in the pancreas in wild type, and the marked reduction of the edema and the infiltrate in the cow mice with pancreatitis. And we confirm that there was a protection against the inflammatory infiltrate in the cow mice because the MPO activity, that's a marker for the neutrophil infiltrate, was also remarkably reduced. And to demonstrate that the upregulation of the transsulfuration pathway was required for the protection in the cow mice with pancreatitis, we performed pancreatitis in the cow mice treated with amino oxacetate, which inhibits two of the key regulatory enzymes of the transsulfuration pathway, which are cystathionine beta synthase and gamma cystathionins. So the cow mice with pancreatitis, without this treatment, they exhibited a reduction in the tissue edema and a reduction in the inflammatory infiltrate. But when we blockade the transsulfuration pathway with amino oxacetate, we avoided this beneficial effect. So the tissue damage, the tissue edema, and the inflammatory infiltrate was enhanced remarkably when we blockade the transsulfuration pathway, demonstrating that the enhanced transsulfuration pathway was one of the key mechanisms to explain the protection in acute inflammation upon deficiency of TRP14. We also, by using immunofluorescence and immunohistochemistry, we confirmed that the inflammatory infiltrate that was present in the wild type with pancreatitis, in the pancreas of the wild type with pancreatitis, but not in the cow mice with pancreatitis, was again present when we inhibited the transformation pathway. So this was that's a critical mechanism for the beneficial effect and for the protection against the inflammation. In collaboration with Antonio Martinez Ruiz, we also assessed and confirmed that TRP14 has a great global impact on the global proteome, has a great impact, and TRP14 therefore may be considered a redox regulatory node that regulates not only cystine metabolism but also the whole proteome. And this would require further studies to discover, to demonstrate which is the impact of TRP14 in different conditions, other inflammatory conditions, other diseases, and particularly in cancer. Our findings have shown that under basal conditions, the cells, when they have TRP14, they have the cystine reductase activity to convert cystine into cystine that promotes glutathione synthesis and protein synthesis, and it's able to reduce protein cysteinylation. When there is an oxidative stress, such as in acute inflammation, if there is TRP14, there is a protection through the reduction of cystine to cystine and the capacity to maintain protein cysteinylation up to a certain level. But if there is deficiency in TRP14, what we have is upregulation. We have paradoxically that the deficiency in the cystine reductase activity causes an increase in cystine levels that activates NRF2, that enhances the transsulfuration pathway, promoting the synthesis of cystine from methionine, promoting the synthesis of glutathione, the upregulation of thyrodoxine 1 through NRF2, and promoting great beneficial effects against acute inflammation. I would like to thank particularly the work by Pablo Martí and Isabella Finamore, who performed most of the experimental work, and of course the collaborations that were essential to perform this work. The collaboration with Elias Arner, Smith, Antonio Martinez Ruiz, Antonio Miranda, and Peter Nagy. And to end, I would like to invite you, as we are working mainly in the redox field, and I am past president of the European Society for Free Radical Research. I would like to invite you to our forthcoming international meeting that will be held in Ireland, in Galway, in June. It will be a pleasure to see you there. Thank you very much for your attention.